foundation laid yesterday in terms of theory, a wonderful introduction to the water cycle by Gabe. And now I'm going to talk about applications. So we're going to talk very broadly about water in biological systems. Microbes, plants, and animals. And I'm going to focus mostly on foundational aspects and not give you lots and lots of case studies. So you've got the handout, so you can probably be just one step ahead of me. I think if we're going to begin to use stable isotopes, we need to be able to answer 4 fundamental questions. Do organisms fractionate when they take up water? Very fundamental, because if they fractionate, we need to understand those processes and to be able to incorporate them. What are the drivers of spatial and temporal variations in the isotopic composition of environmental water? What organisms are going to consume is organismal water. It's environmental water. So, what kind of variations? Gabe gave us a wonderful introduction to what some of those spatial patterns are going to be. Third, what processes influence the isotopic composition of water within organisms? Okay, an organism's now consumed it. What other kinds of processes do we need to be aware of that are going to change the isotopic composition of body water? Whether it's a microbe or whether it's a human. Ideally, there's no fractionation process, so whenever you measure the water inside an organism, it's exactly the same as the environment. That's not the case, though. In no organism is that the case. And then, probably more interesting is that body water, whether it's a microbe or an animal or a plant or a leaf, that body water is going to change over time. I don't want to be Sergio's technician, where I am forced to measure the isotopic composition of that leaf every single minute of every single day during my entire graduate career. No, I don't want that. So what I want to do is to take advantage of Jim's first acronym. And so Jim's first acronym is going to be STIR, which is why I like to use stable isotopes. S stands for source. We can use isotopes as a way of sourcing something, and Gabe talked about sourcing of water. T stands for trace. Let's say I'm interested in the movement of water through a system, whatever that system may be. I will become very apparent tomorrow. That stands for integrate. We can use isotopes to integrate processes. And the R refers to my question number 4, record. So if you think about the fundamental applications of stable isotopes, they're to source something, to trace something, to integrate a process, and to record that process. I'd love to work for Sergio. He's just a fabulous guy to work with. But what a slave driver. I'm not going to be sitting there measuring leaf water the whole time. I want something to measure leaf water for me, and to record it, and to faithfully record it, and to integrate all of the values over the last month. So that's what I'm going to use stable isotopes for. So now we need to ask the first question. Is there fractionation during uptake? And I'm going to make a sweeping statement. But to appreciate that sweeping statement, I need to ask, by a show of hands, how many of you know what an aquaporin is? 1, 2, 3. Okay, half a dozen people. You should learn this. Aquaporins don't even appear in most biochemistry classes. But over time, you will discover this is one of the most important protein complexes. They are PIPs. Can somebody tell me what a PIP is? Plasma integrated protein. So they are proteins that sit in the plasma membrane of microbes, of animals, and of plants. They are most diverse in plants. So every membrane has aquaporins. Now, when I was a kid, I learned that plants took up water by diffusion. And then it diffused across a lipid bilayer. So you see this protein on the right, which is a lipid bilayer. And water is supposed to diffuse across that. Okay? Doesn't happen. Doesn't happen very fast. And then when I was a graduate student in the 70s, another observation was made. If roots take up water and you kill those roots, if it's simply a passive process, they should continue to take up water. But dead roots don't take up water very fast. So I have some facts I need to reconcile. How is it that water is taken up passively? And if it's taken up passively, if a root is killed, it's no longer taken up. Now the answer, which is emerging in plant sciences and in animal sciences and in microbial ecology, or microbial sciences, is that there are proteins called aquaporins. Those aquaporins are composed of a number of different chains, usually 6, and they actually form a little tiny hole through which strings of water, not ions, but strings of water can pass through. So water is moving through the membrane through an aquaporin. The other kind of protein that we all know about are going to be ion channel proteins, which are going to be proteins, they're pips, proteins that are in the membrane, which facilitate the movement of, let's say, calcium, let's say, potassium, let's say chloride, other ions moving across the membrane. And then, of course, we have other proteins that are moving across larger molecules. But aquaporins are a very important part, and we now begin to appreciate that aquaporins take up water. Observation. There are no data today. You might want to earn yourself a reputation by finding those data, but there are no data available today to indicate that aquaporins of any kind fractionate against water. There's no data to suggest that aquaporins fractionate. So that would lead to the fundamental conclusion that there should be no fractionation during uptake by bacteria, by plants, and certainly not by animals, because animals don't allow water to come in by diffusion. They take a massive amount in. They consume the water. But there could be some fractionation across the membranes. But there's no evidence to suggest that, except for 2 studies. Okay. There is a study which was done by Leo Sternberg and his students probably 20, 25 years ago, John? Yeah. In which they suggested that there's fractionation for hydrogen uptake by halophytic or salt-living plants in the mangrove systems. Not fractionation for oxygen, but fractionation for hydrogen. I would love to suggest that that study should be repeated, knowing what we know today. So somebody else repeated that study. Dave Williams and his students repeated a study, and they suggest that there's fractionation. No one else has been able to confirm those studies. Not that the studies are wrong, but no one has gone in to fundamentally ask that question to show that there's fractionation uptake in water. And why do I say this? Because scores of other studies have done the experiment of identifying a certain water composition, adding it to the soil, looking at the uptake by plant, and have not seen any fractionation. So my conclusion at this point is going to be that we have these wonderful proteins and that there is no fractionation during uptake. There certainly would not expect it to be any fractionation during uptake by mammalian systems or invertebrates. In our lab, we have attempted to do this on plants, and we can find no evidence of uptake. We've attempted to do this on microbes, and no evidence of uptake of fractionation during uptake. Okay. So that's going to be my first conclusion. And now I want to be able to apply this, and think about this for a second. And I love working on plants, but I appreciate that most of you like working on animal systems. And you are an animal system. Deer are an animal system. And the water that's available to your environment can differ. So I must recognize that the water source might differ because you move. You drink different fluids. A deer might drink from a stream, might drink from a lake, might migrate. And so if we go to a part of Utah, the Weber River Basin, which is just over the hill, and we look at the isotopic composition of water along that gradient, we will find that there are variations in the oxygen isotope ratio, variations in the deuterium excess. So there is local scale variation in the isotopic composition of water. And just because you might find a difference in the animal time 1, time 2, don't assume that there's a fractionation event. The animal could be migrating and taking a different water source. So within the Weber Basin, which is about a 50-mile basin, we can find variation from about minus 15.5 to minus 17.5. So about a 2 per mil variation in oxygen. And of course, Kate, how much variation will that be in hydrogen? Okay. Anyone want to help Kate? If there's a 2 per mil variation in oxygen, how much in hydrogen? 16. 2 times 8. Okay. Now, let's begin to move a little further into this. If I can get the system to move. Okay. What is that variation associated with? You should say, gee, Jim, I could have predicted that based on what Kate spoke about. It should be related to elevation. It should be something with the cloud moving up and releasing precipitation. As temperatures decreases, the cloud increases in elevation, and you see, lo and behold, there's a very tight correlation between the water we sampled and the isotopic composition of water and the elevation. So the top of our sampling was someplace around 2,500 meters. Down in the valley, we're down around 1,500 meters. So we have about a 1,000-meter gradient, and we see variation in the isotopic composition of the water, and we see variation in deuterium excess, exactly as you would predict. Okay. So we have variation locally. So if you're studying animal systems where movement is important, recognize that you get that variation. Of course, that doesn't apply to plants. So let's stop here for a second.